In this video, we will discuss about expression of structural and accessory proteins in case of SARS-CoV-2. So, what are structural proteins? Structural protein are main proteins that we have discussed. That is membrane protein, nucleocapsid proteins, spike proteins, and envelope proteins. And they are mainly found in open reading frames of SARS-CoV-2, and interspaced between open reading frames are the accessory proteins. Accessory proteins are not conserved, and there is lot of variability occurs in accessory proteins of SARS-CoV-2, whereas in other proteins they are found to be more conserved than in SARS-CoV-2. So what people have right now hypothesis that this the accessory proteins of SARS-CoV-2 might be required in natural host, wherever it has evolved, but it is not that much required for the host on which it is targeting, not required for affecting host. There are general ORFs which are responsible for accessory proteins are usually found in ORF 3A, 6, 7A, 7B, and there are some few other ORFs, that is 3B and 9B, that are usually found when there is a leaky reading of sgRNA that we have discussed in the previous last video, that sgRNA might continue to read even though the translation end signal reaches to sgRNA. So the main thing when the SARS-CoV-2 initially affected the human beings, that is during the initial time period of pandemic, this recent pandemic, people have found that ORF8 is present, or it is translated completely, and this ORF8 and in SARS-CoV ORF3B, 3B, ORF8 and ORF6. These three ORF, the number might need to reconfirm it. You can confirm that in the reference, that is Vinachai. But in SARS-CoV-2, 3 ORF are responsible for antagonistic interferon activity. That is, they are responsible for taking care of interferon that is being generated when SARS-CoV-2 affected the cell. And in this case of SARS-CoV-2, that function has been taken care by ORF8 and other ORFs also. ORF is open reading frame. So in this ORF8, initially it was found to be completely translated. However, later on it was found that some of the sequence got deleted with time, and the sequence has been deleted to that extent that ORF has been now divided into two, translated in two fragments, ORF8A and ORF8B. and there are some slight deletion. A deletion is also observed. There are two lethal deletion for ORF8 observed. Lethal deletion for ORF8. It observed. That makes this ORF8 infunctional, non-functional. And I think I have spell the meaning of antagonist interferon activity. This means that interferon has been able to interferon has been able to affect the SARS-CoV2 whenever the antagonist interferons has been detected by the cells. So the interferons are generated when they see antagonist interferons, that is this kind of ORF. When translated, then cell's immune function will be able to function, cell's immune system will be able to function well. Whereas when this in case of SARS-CoV2 with the deletion of ORF8, it is now been considered as been considered as immune evasive strategy by the virus, since this ORF is not been functional. It is not been detected by the human cells or any cells for which SARS-CoV2 affected, and because of this, interferon activity has not been activated to that extent. So this can be considered as one of the strategy by SARS-CoV2 virus to make it less pathogenic in the cells in which it is affecting. So that's all for this expression of structural and accessory proteins of SARS-CoV2. Thank you.